in the human genome, it's only about 1.1 to 1.4% that is actually going into translation of protein product. So what happens to the rest? People thought it is a junk DNA, and people also call that as dark genome, because once you do not know something about, you call that as dark, you are in the dark. So that is the idea. This is what we have said that yesterday, and some of the other things I have mentioned, transposons, how much is RNA, how much is the duplications, and so on. And we also compared the genome of chimpanzee with that of humans, and more difference can be seen in the non-coding regions, about 4%, whereas coding region it is only 0.1%, approximately only 20 genes are different. So what makes the difference is the non-coding region, not the coding region. So that means there must be some function to that. After all, the basic idea of epigenetics is that I already mentioned that we are all born by the fusion of sperm and ovum, and what you get as a zygote, which looks very different from the fetus, where some morphological changes are seen. But then the genome content is same in all the cells in the fetus also, whether it is a liver cell or something. So that means something is there above genetics. Genetic refers to the genetic information, right? So the genes are same in all the tissues, but something is more than that. So what is that? We know now it is gene expression. So there must be some control without changing the genetic content, because the genotype is same but phenotype is different. So that is the idea behind all these things. So, so there are other examples also like that. So junk DNA, finally they found that there is a role in gene silencing, most often as post-transcriptional silencing of active genes by what are known as microRNAs, siRNAs. They are about 21 to 23 base pairs. MicroRNA can bind complementary mRNA and stop transcription. So for these related discoveries, there was a Nobel Prize in Medicine and Physiology in 2006. Andrew Fair and Craig Mello, they are from the US, specifically somewhere near Boston, UMass they call, University of Massachusetts. So they belong to that university, and they discovered this. So they were given Nobel Prize. It's not that they only discovered, but they made the major contribution. But much before that, before these people contributed, somewhere in 1990, Rich Jorgensen at the University of Arizona, his group was trying to deepen the purple color. You see, mainly it was from plant work. They have done some work in plants, flowers and all that, and that ultimately the knowledge gained from that has expanded to humans and other organisms. So they were trying to deepen the purple color. You see the purple flower here. So they thought this is not that deep, so let us make it somewhat darker. So they introduced the gene responsible for this. This is known as a petunia flower. So petunia, but were surprised at the result. What happened? Instead of that, they got something like this and something like this. They wanted to deepen the purple color, that didn't happen. Here they have more than one color, and here you have almost everything is white. So they thought that this is due to some suppression of the expression, because they were trying to express something, but that was not coming. So they thought that is somehow getting suppressed. So they called it as co-suppression. Since the expression of both the existing gene, the initial purple color, and the introduced gene to deepen the purple color were suppressed, they called it as co-suppression. So co-suppression has since been found in many other plant species and also in fungi. It is now known this is all due to double stranded RNA. Now we know that but this is the initial discovery. This formed the basis for a later discovery. But what I am going to show you now is the most important experiment that led people to believe that it is double stranded DNA, I mean RNA, that is responsible for this kind of a suppressive effect, or in other words gene silencing effect. This is a C. elegans, you know C. elegans what it is, that embryos, once they added antisense for a particular RNA, they got somewhat this kind of a color, but when they added double strand RNA, they completely took out this color, it got bleached. You may not understand at this stage, but look at here. This is again C. elegans, this is the worm. So, you have a sense RNA, this is all for a particular gene which is called UNC22, UNC22 RNA into the gonads of C. elegans. They have injected single stranded or double stranded RNA for this gene, and the UNC22 gene encodes a myofilament protein. Decrease in the activity, suppose this gene is not there, the worm will be twitching. So, their idea is what can make this twitching, in other words what can suppress this gene, that is the idea. Now, they have given antisense RNA, that is single, the sense RNA, antisense RNA, that is single stranded, both are single stranded, either sense RNA or antisense RNA, single stranded, whereas here they have given a double stranded RNA. So, only in the double stranded RNA when it was transfected they saw twitching. So, they thought that double stranded RNA but not single stranded RNA induce the twitching phenotype in the progeny. So, this is what they found. So, they, in other words, this was one of the first discoveries to show that the double stranded RNA can silence the expression of a gene. So, this was done like that. Later on, of course, the these are known as non-coding RNAs because they do not code for a protein product, there is no protein product. So, they called it as non-coding RNAs, several of them. Several small RNAs have been discovered. The famous among them is these days microRNAs, miRNAs. There is also what are known as siRNAs, snow RNA, piRNA and so on. So, a lot of such discoveries have been made over the years. What we are going to talk about is microRNAs. So, we cannot discuss everything. So, we will go one by one. How the microRNAs were discovered? We will go by the historical account first and then we will study the functions of the microRNAs, what are microRNAs, why we are interested about microRNAs in cancer biology, all these things we can build up a story on that. So, one of the first discoveries was that LIN4 is a gene, it was responsible for degradation of let us say it codes for a protein LIN4, it is responsible for the degradation of protein called LIN14. Mutant worms deficient in LIN4 function had high levels of LIN14. Hope you understand. If LIN4 is not there, there is high level of LIN14. That means LIN4 somehow suppresses LIN14. So, that is the idea. In 1993, I think in New York, Ambrose and colleagues, they found that LIN4 did not encode a protein, regulatory protein. First thing is this is a gene, but it does not encode any protein. So, that is what they found. Instead, it gave rise to small RNA molecules of about 22 to 61 nucleotides. Since one was small, they call it as LIN4S, since the short LIN4, 4S means short, and the 61 nucleotide one was called long. So, they called it as LIN4L. So, this was the naming. The sequence analysis showed LIN4S was part of LIN4L. In LIN4L was predicted to form a stem loop structure with LIN4S contained in one of the arms, so called 5' arm. What is more important, forget all that. Lin4s was partially complementary to several sequences in the 3' untranslated region of the messenger RNA encoding Lin14 protein. Now we all know that microRNAs bind with the 3' UTR of the target mRNA, but this was the first discovery. Here the target is Lin14 and the RNA is Lin4. So, Lin4 has complementary sequences. So, that is what Lin4 binds with the complementary sequences that are found in the 3' UTR of the Lin14. Sorry. So, they hypothesized that Lin4 could regulate Lin14 through the binding of Lin4s, the short sequence, to these sequences in the Lin14 transcript in a type of antisense RNA mechanism, something that is complementary and that ultimately degrades it, something like that they have hypothesized. They were not very clear at that time. For nearly 7 years, nobody bothered about this. Although it was an important discovery, I will tell you why they started bothering about this. In 2000, another C. elegans small regulatory molecule called LET7, this is a microRNA, LET7 was characterized and found to be conserved in many species. This is the most important thing. Once they came to know that this mechanism is there in every organism, suddenly the interest develops, because it will be useful to the humanity to treat the disease or something like that. This is how it was. That is why somebody said, in evolution classes people used to teach, someone said that nothing in biology makes sense without connecting to evolution. They said that. So, this is one example. Without connecting to other species, something that you discover in some lower organisms, people may not be interested. They will think just like this junk DNA that I told you, people think that this is junk information, although it may not be junk. In biology, that is the beauty. You know the difference between discovery and invention, right? I hope you know. Discovery is made out of something that is already existing. That is what is existing in nature. Nature keeps it covered, you are uncovering it. That is what that's why these kind of discoveries in biology, you call that as discovery, not invention. Whereas in the engineering side, if you alter some machine and all that, you are going to say you have invented something, some new machine, something like that. So, that is one important difference. Why I said yes. So, till other people come in and say something, people do not accept that or they ignore that and things like that. So, in 2000 they found this LET7. These discoveries ultimately confirmed that Ambrose had in fact discovered a class of small RNAs with conserved sequence and functions, and these molecules now we are calling them as microRNAs. This is the discovery. As I mentioned you, the other portion of the genome where they had no idea, that is non-coding region, they called it as dark genomic matter. MicroRNAs, now we know microRNAs are also non-coding RNAs after these discoveries. So, they have to reconsider this term dark genome, because this is not dark anymore, some light is being shed now and we start understanding more about that. That is the idea. So, microRNAs, what are microRNAs? They are small, evolutionarily conserved, non-coding RNAs of the length you have to mention 18 to 25 nucleotides. This is a typical definition. What is their function? They regulate expression of genes. They are expression regulators of genes. What do they do? What do the genes do? Anything that you can think of, the cells do. Fundamental cell processes. What are the fundamental cell processes? They are involved in development, because organisms have to develop, right? From embryo they have to develop ultimately into full organism. Whatever, development, differentiation, proliferation, survival, death, you name it. You name any function, you will find microRNAs being involved. So that's why people say small but have important function. They are tiny regulators, but their functional importance is not tiny, very big. That I will also tell you, there are so many microRNAs in the system. It keeps on changing also as you go back. I guess it's about 2500 microRNAs are there in the human genome. If you take with other organisms, it can be 5000, 6000, but in human genome about 2500 microRNAs are there. The most interesting thing is the microRNAs, they silence the gene expression. So the gene that is getting silenced is called a target. So you might ask whether one microRNA will have one target. No, they will have more than 500 targets. Interestingly, the target, one target may be targeted by many microRNAs also. So one microRNA can have many targets, one target may be targeted by many microRNAs. So this makes a complex story there. It's not easy to understand because they are going to regulate 500 genes each microRNA, and you have 2500 microRNAs in the system. So you can understand the complexity. In addition, these days we are also hearing more about what are known as long non-coding RNAs, sometimes called link RNAs, LNC RNAs. It does not stop there. There are also circular RNAs. Some of the RNAs are circular. So apart from the DNA, there are thousands of RNAs in the system which do have regulatory roles, and we do not know the functions of all of them. It's very difficult because now we are talking about thousands of RNA molecules. So RNA biology has emerged as an entirely different field now. People are working, a lot of people are working on that. Now we will come back to microRNAs. MicroRNAs are just like any other RNA, they have their own genes. Almost 60% of them have their own genes. They can be transcribed from intragenic or intergenic regions by RNA polymerase II, just like any other RNA. So the primary transcripts are usually synthesized as lengthier molecules about 1 kb to 3 kb, which we call as primary microRNAs. They are again spliced, or they are streamlined, processed further by certain enzymes called Drosha and Digeorge syndrome critical region 8. It is called a DGCR8. This is a complex present in the nucleus, and thereby it can process this primary microRNA into a hairpin intermediate about 70 to 100 nucleotides called pre-microRNAs. So you have primary microRNAs, you have pre-microRNAs. After that, you have the real microRNA. Before that, I will tell you, the targeting is usually on the 3' untranslated region of the target gene. What is the length? About 2 to 8 nucleotides. This is often called a seed sequence in the target. That is what is targeted. Targeted means they are exactly complementary. The same Watson and Crick complementary, A base pairs with T, G base pairs with C. So once you know one sequence, you can predict the other sequence. So once you know the 3' UTR sequence, you can predict what microRNAs will target that. So accordingly, number of bioinformatic programs have sprung up. So many bioinformatic tools are available to predict the microRNAs that can target the genes of your interest. Whatever is the gene that you are interested in silencing, what you will be interested to silence in cancer biology? You want to silence the tumor suppressor genes or oncogenes? oncogenes. So, you want to do that, or you want to express the tumor suppressor genes, right. So, accordingly we can play it up with that. I will come to that a little bit later. So, when the microRNA and its target mRNA sequence show perfect complementarity, that is called 100% complementarity, then the mRNA gets degraded, no protein product. Interestingly, most of the plant microRNAs are like that. In the plant there is 100% complementarity. In the animals it does not happen, it is not 100%, usually there is a gradation. This gives rise to some scope of some this is also related probably to evolution, some complexity in the expression. But ultimately here also, even if it is partially complementary, the mRNA will be ultimately degraded, or there may be inhibition of the expression of the protein. Ultimately what is happening is the protein product is not expressed, whether it is partially complementary or whether it is fully complementary, but fully complementary the mRNA gets completely degraded. So, that is one important difference. Since I told you that is what the net result is, a decrease in the amount of proteins encoded by the mRNA targets. So, that is that I have given it in red. Each microRNA, what is important is each microRNA has the potential to target a large number of genes, about 500 for each microRNA family. So, this is a very important thing to just remember, because you see in cancer we need such molecules, because cancer is a disease that where you have aberration of many genes, multi genes. So, something that can block multi genes will be of some use to cancer. We will again come back to that. So, about 60% of the microRNAs have one or more evolutionary conserved sequences that are predicted to interact with microRNAs. Bioinformatic analysis predicts 3' UTR of single gene is frequently targeted by several different microRNAs. This is something that I just told you, I just read out. Now, once the pre-microRNA is formed, we have talked about primary microRNA and then pre-microRNA. These are all happening in the nucleus, right. So, this has to go to the cytoplasm. That is done by what is called exportin 5. It is a mechanism of transport through exportin 5. In the cytoplasm, the pre-microRNA is further processed by another ribonuclease called dicer. This is a very important thing you must remember, into a mature double stranded miRNA of about 18 to 25 nucleotides. Now, it becomes double stranded. After the strand is separated, you have two strands now, and if they are separating out, one of the strands is going to act as a microRNA, that is called a guide strand or a mature mature microRNA you want to call it. The other strand is called passenger strand. Passenger is not driving, right, he is only just watching how the bus goes, something like that. So, it is not doing anything, but this concept is changing these days. Usually it is thought that the passenger strand, people earlier days they were representing it as microRNA star, but nowadays there are new theories and some experimental proofs to show that both the strands can act as microRNAs. So, one is called 5' microRNA, the other one is the 3' counterpart of it. So, when they give the names for microRNAs, they have devised like that the 5' strand and the 3' strand, something like that they say. Both the strands may act, but generally this is the case, the passenger strand gets degraded and the one that acts as a microRNA is called guide strand. There is a complex which is called RNA induced silencing complex, RISC complex of the microRNA pathway is comprised of microRNA, and there are other proteins what are called organoid proteins. There are about 4 proteins in the mammalian system. organette 1, 2, 3 and 4. So, these protein factors are also important. Organette proteins have a crucial role in the microRNA biogenesis, maturation and microRNA effector functions. The mature strand is important for target recognition and for the incorporation of specific target mRNA into the RISC complex. That is what, anyway I will show you one of whatever I have discussed, you can see in this. Like any other microRNA, RNA polymerase comes in and you get a primary microRNA. You see that, do you see this poly A tail and all that, and this loop, all these structures are that there is a 5 prime cap, just like any other any other RNA. And then once the drosha and GGCR8 comes, this lower portion is cut off. You can see that here. Are you able to see that? Pre-microRNAs. Here you are seeing this 5 prime cap and the poly A tail, all those things. I hope you know the stem loop RNA structure, you must be familiar with. So, this is called pre-microRNA, exportin 5 mechanism. Now, we are talking about cytoplasm. So, the dicer comes in, the RISC complex. So, you get the double stranded RNA, and then there are lot of transcriptional ribosomal binding proteins, lot of them are there. That is what I told you, organette and all that. And finally, there is a separation of these strands, and one of the strands will be binding with the target mRNA in the RISC complex. So, this is what is the so called microRNA biogenesis. So, how microRNAs are formed? It is not as simple as that, there are also some complications. This is about microRNAs being formed from their own genes, that is they have a DNA of their own and they form just like any other microRNA. But sometimes they can be formed from introns also by splicing. So, that is also possible. So, as I told you, majority is by this method, 60%. So, some microRNAs could be derived from intronic regions also. They are sometimes called mirtrons. There are different names. You can see that here some of the, this is what mirtron gene is called. So, you can have transcription, this is one way, this is the usual way that I mentioned. Here there can be spliceosomes and the excised introns can ultimately form a small microRNA. So, by basically by the spliceosome mechanism, from the intronic region also you may get some of the microRNAs. So, this is basically explained here, that is this part is called the first pathway canonical microRNA. Canonical means the usual way, and this is the mirtron biogenesis. As I told you, those which are formed from the intron are called mirtron. This is the nomenclature. Now, we are coming to the important aspect of this, that is why we are studying in cancer biology. This is also more or less same figure only, it is just more clear. You will see that there are also sometimes there are some RISC independent functions also. The miRNA may bind with sometimes with other DNA or even with other RNAs and so on. So, they can bind with even DNA sometimes in a RISC independent fashion also, and some proteins also may bind with them. So, those are some different functions of that. Sometimes they may bind with 5 prime UTR also, these are deviations. Let us not worry about that. General reaction is the microRNAs will bind to the 3 prime UTR in the complementary region and usually result in gene silencing. There are some occasions where they can bind in 5 prime UTR and can activate gene transcription also, but that is an exception. Do not worry about the exception. Now, the other interesting part is, since I told you that they are formed from genes, whatever the gene aberrations that can happen to microRNAs also. That is, their expression can be altered by mutations. You can see here that miRNA promoter, it can be regulated even by methylation, for example. The miRNA producing gene may be silenced by hypermethylation of its promoter, epigenetic regulation, it is possible. Or alternatively, the gene may be mutated and the microRNA may not be expressed. Or there can be single nucleotide polymorphism, SNPs, deletions, amplifications, chromosome translocations. All these abnormalities can affect the microRNA producing genes. Remember, 60% of them have their own genes. So, those genes can be regulated by the regular mechanisms. So, there can be mutations and all. In this way, the expression of it is one thing that the microRNAs regulate the expression of several genes. It is another thing, the microRNA expression in itself can be regulated by aberrations in the genes that are responsible for the synthesis of the microRNAs. Do not get, I hope you are not confused about it. So, that is important for you to understand this. So, it is a tightly regulated process, it is having multiple steps. So, this is what, again the defects can be anywhere. Here there is another thing, the defect can be in the drosha, for example, or in the risk complex there could be some defects, or in the base pairing there could be some defects. So, the deviations can happen in any of the steps that are involved in the microRNA biosynthesis. So, this has to be kept in mind for understanding certain alterations in many diseases or pathological conditions. That is what is shown here. See here, sometimes you may have drosha downregulation or the exportin 5 mutation and so on. So, it can result in certain microRNAs, certain set of microRNAs. For the purpose of cancer biology, we will discuss what are known as oncomers, means oncogenic microRNAs. There are also tumor suppressor microRNAs, or you can call them as anti-oncomer, you can say that, but usually we say tumor suppressor microRNAs. So, this is why we are interested in microRNAs, that is, the microRNAs can behave as oncogenes or tumor suppressor genes. So, that is of immense interest for cancer biology. Now, you may ask, sir, how they can behave as oncogenes and how they can behave as tumor suppressor genes? How is it possible? Simple. You remember the rule that I said, if you suppress the suppressor, you activate something. If the microRNA suppresses a tumor suppressor, microRNA function is to suppress the gene expression, silencing the gene expression, right. If it suppresses a tumor suppressor, it will be oncogene, oncomer. So, oncomers will have a target as, what will be the target for oncomer? Tumor suppressor genes. Those microRNAs which target tumor suppressor genes are called oncomers. Reverse is true. The tumor suppressor microRNAs, they will target oncogenes, they will suppress oncogenic microRNA. Simple. It is very interesting. This is the main reason why we are interested in microRNAs. It is also equally interesting that there are only two strategies that we look for. You remember I told you, anything we learn in cancer biology, ultimately we want to make use of that knowledge to develop treatment, to develop strategies for treatment. So, what will be the treatment by using this? We know now, we have learned that they can behave as oncogenes, they can behave as tumor suppressor genes. What is the use of this? How will you make use of that? If you know that in the patient there are oncogenic microRNAs you want to inhibit. That is known as microRNA inhibition therapy. There are only two therapies. And if you want to introduce tumor suppressors, you want to introduce this tumor suppressor microRNAs. So, that means the patients are having very low levels and now you are introducing tumor suppressor microRNAs. So, in other words, you are replacing them because it is not there, it is only in minute amount, you are replacing the deficiency. It is deficient, you are replacing them. That is called replacement therapy, microRNA replacement therapy. There are only two therapies, microRNA inhibition therapy, microRNA replacement therapy. But there is a catch. This microRNA inhibition you can do by 1000 ways. MicroRNA replacement therapy you can do, you can have 1000 strategies. You can have so many different ideas to do that. Basically, they fall in line with these two broad strategies. Ultimately, whatever is the novel method that you are discovering, you are ultimately doing one of these or both. You want to do both in some cases to be more successful. So, this is, if you understand this, you understood. Now, how this can happen, how these oncomers can form and how tumor suppressor microRNAs can form is shown here. You see, due to these defects, sometimes there can be more than the normal copy of the oncogenic microRNAs. That is what is shown here. By aberrations in these procedures, for example, if there is a mutation or if there is an amplification, over amplification, the particular gene may be produced in excess. So, that will result in accumulation of oncogenic microRNAs. Of course, loss of tumor suppressor microRNAs, all these things, it can happen because if there is a defect, then it can be loss of tumor suppressor microRNAs. So, these two mechanisms may account for the accumulation of oncogenic microRNAs. That is, either you can have accumulation of oncomers or loss of tumor suppressor microRNAs or both. If these things happen, then the net effect will be production of more oncomers. Do you catch this point? You do the same thing. You suppress the tumor suppressor, you increase the oncomer, do both to get a net result of getting oncomers. So, that is what it is. This is an important slide. You see here, now that we have understood what is microRNA, and remember, although the microRNAs are 18 to 25 nucleotides, we are talking about the first 8 nucleotides, which is called a seed sequence from the 5' end. Always the counting, the number is from the 5' end, right. So, that is called a seed sequence. Now, once the microRNA expression goes up, whatever is the target, what will happen to that expression? It will go down. That is what is shown here. For example, during development, if a particular microRNA gets synthesized over time, at the same timeline, the target expression will go down, correct? This is known as a temporal switch. I have given some example, Lin4, Let7, all those kind of things. I told you these are some microRNAs and the targets also I just told you. Lin14 is the target, right? So, Let7 targets there are many. So, like that. Now, this gives rise to very important concepts. Why we are talking about this microRNA, what is the importance? We are just saying that they are silencing the gene expression. What is the big deal? There is a big deal, because you remember this example, once again I am telling you, liver cells, kidney cells, all of them have the same genome, but different levels of gene expression, because they express a different set of genes. So, if you control gene expression, you can even convert a liver cell into a kidney cell, theoretically, right. So, that is very important. Cell type can be changed. If you alter the expression pattern, you can probably convert one cell into another cell, cell type A into cell type B, that is what is shown here. So, these are mechanisms that may be responsible for the formation of different cell types. For example, in this case a microRNA is off, that means the target is on, right. We need for this cell type A, this particular target should be on, should be expressed. Now, in another cell type B, we want the target to be off. So, in that cell type the microRNA is on. So, the on off switch for microRNAs plays an important role in cell type formation. That is what I am trying to say. This is known as spatial switch, they call, same thing. Now, there is another thing, it can help you, it is the same thing basically. What we are saying as the spatial switch is almost the same like this one. This helps in tissue identity, that is, if microRNA can target many genes and if it particularly targets some set of genes, that it can help in identifying a tissue, because in that tissue we know that only particular set of genes have to be expressed. If the microRNA does that, then you have a microRNA that determines the identity of the tissue, correct. Theoretically, it is not going to be done by one microRNA, it is not going to be done by something, because if that microRNA does this, there is an opponent, some other microRNA which may undo this, that is also there, do not forget that. There is always a competition between the two. Who wins will determine the cell identity or tissue identity, that is one thing. Now, this is very important, because if the cells want to get transformed into cancer cells, they want expression of certain genes. So, repression of many targets whose expression is inappropriate for tissue X. For a cancer cell, what is inappropriate is tumor suppressor, it does not want the tumor suppressor. So, it will silence that using these microRNAs. So, when the normal cell gets transformed into a cancer cell, it will see to that it will express these microRNAs which will shut down the tumor suppressors. So, it helps these cells to get transformed into a cancer cell. Whenever I am explaining this, do not think this is the only mechanism, it is never like that. There are thousands of similar mechanisms that are happening, but under some conditions these mechanisms may get a upper hand. So, that is how you have to understand. Never think that when we are explaining some mechanism, do not get into a mechanical mood where only this one will do that. It is not like that in biology, I want you to keep in mind. So, fine tuning the expression is what ultimately we are doing. So, when a microRNA is on, modulation of many unrelated targets also possible, because there are 500 targets. So, it is quite possible that you may be blocking many unrelated targets also. So, these are all the important things that you may have to remember. This is another very interesting story. When we were doing all these experiments, you know, when I joined here, this is what, June 2010, yes, there was a paper in Nature. We were all excited. See, when I came here in 2006, I came here from Rajiv Gandhi Center. There is no rule that you have to do only this kind of research. In institutions like this, if the PI thinks, if the professor thinks that he wants to do something, I took something about which I knew nothing, that is microRNA. At that time when I came here, I did not know about microRNAs. I only know that it is something new area that is coming up. As some students also were interested, so I thought why not we start something, because I was doing on NF kappa B signaling, you remember, I was just telling NF kappa B signaling, TGF beta signaling, because I was in Trivandrum, I came over here. Of course, at that time itself, I was a little bit more than 50 or something. So, anyway, I have done something there and I wanted to start something here, though late, but so we started this. So, I had no idea about microRNAs. All I could understand from the literature is there will be a complementary binding, that attracted me, because these BTech students here are very good in maths or playing with computers and all, which I was not. So, I thought one way to do this is to ask some student to find out some computer program that will find the complementary targets for a given microRNA. One project student was interested. This is how I started actually. One project student was interested. So I just told him, come on, go, and I don't know what is this software and I don't know how to use it. So if you know, you come up, we will make a project. This is what I just told him, and he was very interested. Next day he came up with one, he took some random microRNA and came up with some 100 targets. I told him, come on, we have so many 100 targets. How do we work with 100 genes? It's not possible. So then I told him, look, I have some track record, I have worked on NF kappa B, TGF beta and all that. You take those genes, because we have some reagents in the lab. So you do that, and then you look for microRNAs that can target. See, if you can do by complementarity, if you can find out the microRNAs that can target a particular genes, from the target sequence, why don't you go back to the microRNAs that will target this, right? It should be possible. Now it is not a new thing, but at that point of time, not many were doing also, like they were just, it was coming up. So I just told him. Next day he came up with again bunch of microRNAs that will target. So I was a bit worried how to start working, because see, you are starting with an open mind in a research area of which you don't know also much, and so how to start. So that was my dilemma. So I asked him, what is this? Again you are coming with so many bunch of microRNAs that target. So what will I do? I don't know about any of the microRNAs, so it doesn't matter what I start with. Only I know about the gene NF kappa B or SMAD, TGF beta, because I have worked on that. So I told him, okay, we will do something on SMAD targeting microRNA or NF kappa B targeting microRNA. He came up with some names. I looked at it, I looked at the literature, and called up some friends abroad and told them whether they have something that can give me an expression vector, because I don't want to do it. So I asked them, so they provided some microRNAs. Very interestingly, what we thought in bioinformatic methods, this fellow also brought me some scoring. It gives a scoring depending on the binding, depending upon the different conditions, that is, what do you call that, binding constants and all that. He came out with a score that will give a very high score for binding this microRNA. So I thought, here we got, for this microRNA, this is going to be the target, isn't it? Very interesting. So I got the microRNA expressing vector. I asked the students, now I turned to PhD students, because he was only a BTech student. That project was over, I sent him out, and I asked my PhD students to do this transfection, because we were doing transfections all the time in some cancer cells, and then come out with antibodies for that target and find out. The first thing you will do is you transfect the microRNA, and then you see the target miRNA as well as the protein. What should happen? It should decrease. The mRNA you are going to look at by RT-PCR or PCR, whatever you want, usually RT-PCR, and then the protein you are going to do by Western blotting. I hope some of you understand. So I looked at it, nothing happened. What we thought as the best microRNA, we wasted 2-3 years, nothing happened. So it basically tells you the biogenesis. Like I told you, it can come from its own gene by RNA polymerase II, and it can be intergenic, or it can be from, I mean, like introns, or simply splicing tRNA, refolding and all that. It can ultimately result in pre-microRNA, which can be converted to the so-called primary microRNA, and then you have pre-microRNA, which can be transported to the cytoplasm via this complex, what is called exportin 5 RAN GTP complex. And if there is a high complementarity, then you get degradation. Where is it shown? Here you can see it's translational, ultimately translational inhibition. If it is very high complementarity, there is mRNA degradation. that is happening ultimately in the risk complex where I told you about dicer and the several proteins, RNA binding proteins. It's a complex. With low complementarity, you get translational inhibition ultimately. Apart from that, it can also bind RNA. There is one more thing that I did not say, but it is seen in this slide, is this: the microRNA can also enter what are called exosomes, and they can come into this through the process of exocytosis and all. It can find its place in the exosome. It can be carried on. So there are some other deviations